It's real. So I'm going to explain the basics of that. And after today's lecture, you're going to be able to walk away and go, I know everything I need to know about genetics, genome engineering, and stem cells like this. And then what I'm going to do tomorrow is, and I'm going to mention a couple of examples at the end of today, but then tomorrow's lecture is trying to give a more African sort of perspective. You know, why should we be doing what are considered really two of the most advanced biological technologies in a country with such a massive health burden and all the other issues that we've got? And I'll mention that, and then I'll talk about some of the ethical things as well. I'm weird with microphones, so if they start making funny noises, can someone just put their hand up and go, it's done something? Yes? Thanks. Okay, so by the end of tomorrow, you will understand this diagram perfectly. Okay, and it looks a little busy now, but really what I want you to be able to do is, because there's so much information, it's an informative lecture where when you see things in the newspaper, or BBC does another article about something, or CNN posts something, you can look at it and you can go, I know which method they used, and that didn't make sense, or oh my goodness, now I realize why they're in trouble, or okay, that sounds pretty much right, because if I remember Janine telling me the right things, then that's kind of the way you can do it. So genome engineering is complex. You have various ways in which you can use it. So I'm going to get you to understand by the end of it the way that you can directly add genome engineering tools to a person as a treatment. I'm going to explain to you how you can take cells from individuals, from every single person in this room if we wanted to, engineer those and possibly put them back into a patient. And then I'm going to talk to you a bit about how you can take similar stem cells and actually use them for modeling disease in a dish. And this is really where my work sort of fits in. So those are the three different avenues. So let's just talk about why therapeutic genome engineering is important. There is a well-established history of cellular transplantation treatments. Bone marrow transplants have been optimized for decades. So that is a cellular transplant treatment. It's a cell therapy. But the problem is that we're moving also from just the cell therapies of that kind, hematopoietic, the blood ones in particular, to a massive need for organs as well. There just aren't enough organ donors. So we need to start trying to figure out how we can make more of these in a dish in the lab. And this is also where these kinds of techniques come in, so we'll talk about that. There's one particular phrase that's going to come up quite a lot, and that is gene therapy. Genome engineering is a tool that can be used for gene therapy, but it has one specific advantage. It's targeted. It's very, very specific to a certain part of your DNA. And I'll explain that a little bit more. So it falls under the umbrella of gene therapy, but uses a very specific tool. Why is it so hard? Because it uses everything from your lab rat geneticist that I once was through to your most advanced surgeons and medical personnel with myriad health practitioners in between. You have immune problems, immune rejection problems. If you're going to add something foreign to a body that's of a biological nature, your body, if it's doing the right thing, is going to respond and say this isn't right. Sometimes, though, your immune system isn't working hard enough, and the cells, for example, that you add overtake, that's also bad. So it's this delicate balance that you've got to figure out with these techniques as well. So it's complex, it's complicated. But given all of these problems, it really is going to be able to change the lives of people around the world for which we never thought it was supposed to. And this is perhaps the best example. So I don't know if any of you saw this a few years ago. This is Leila. When she was 6 months old, she was at Great Ormond Street, and she'd been diagnosed with a very severe type of acute myeloid lymphoid leukemia. Acute leukemia, sorry, I beg your pardon. And the doctors, her team, were done. They'd tried absolutely everything they could for this little thing. And then one of her team members recalled that a French company called Selectus had genetically engineered some T cells. They're a type of blood cell. In fact, if cancer is occurring in your body, your blood cells actually go, your T cells go directly to that cancer and supposed to chop it up. That's part of how the immune system works. But she didn't have very much of an immune system left, and hers weren't doing what they were supposed to be doing. But what this French company had done is they said, okay, well, we've got these T cells, and we know for this type of cancer that there's a specific marker, there's a little antigen on the surface of these cancer cells called CD19. So what they did was they then made the receptor, a gene that codes for a receptor specifically for CD19, and engineered it into the T cells. Now they have cells that they could give Leila, where you infuse these cells and then rush directly to all of her cancer cells and destroy them. But there was a problem. And the problem is that the T cells weren't hers. So if they gave them to her, there'd be an immune, like a host graft disease issue. So there'd be an immune rejection. So the doctors were like, okay, cool, that's fine. We've got the one genome engineering thing, so these will be super cancer targeting cells. Let's do a second hit, a second genome engineering modification. And they disabled that recognition gene in these T cells so that it couldn't be recognized by her body as being foreign. They blinded it. And they infused them into her. They did 2 treatments. This photo was taken 6 months later. She was a year old. She had another treatment in March in 2016, and she's been effectively cured of this. She was the first. There's a second little girl done a year later, same treatment, same team, if I'm not mistaken, at Great Ormond Street. In a rare situation for scientists, they were very not too excited about what was going on. They just said, look, it's just 2 patients, so let's not go overboard. But as of March this year, this is called CART, chimeric antigen receptor for T cells, has been approved by the FDA. So if you have $468,000 and you have this type of leukemia, then you can be treated with this. But this is the magic of genome engineering. This has taken something that was not treatable at all. There was nothing left that they could do to something that very realistically could actually be an option. And like everything in technology, the first thing that you make, the first pill costs $1 billion, the second pill costs 10 cents, right? It's in the research and development. So that's Leila. She's like the flagship of genome engineering. So given now that I've got you very excited about what this can do, now we have to go back to the basics. And so I want to talk to you about how we can actually do genome engineering. Some people call it editing, genome editing. I'm going to talk to you about the different cells that you can use. Your body has just got all these incredible cells that you can manipulate for different reasons. I'm going to talk to you about delivery and mention something about a de-weaponized tank. And then briefly towards the end, I'm just going to mention a couple of examples, and that should get you started for the basics. So forgive me if I'm being patronizing, but I realized as I was putting this together, if I'm going to talk about how you have to chop up genes and genetics and DNA, I have to explain what a gene is. And very simply, we've obviously got billions of letters of code in every single cell in our body, and a gene is just simply a functional element of that DNA that codes for something. It doesn't have to be able to turn into protein or even RNA, it just has to be functional. It can just be the sequence itself that's important, and I'll explain that in a second. So it's the blueprint, right? So we have billions of these in our cells, and the gene is just the one that's just a simple functional element. And thanks to the Human Genome Project, we now know what a lot of them do. We actually are increasingly realizing we know less and less than we thought, but we know what a lot of them do. So what's important about genome engineering when it comes to this? So the central dogma, Watson and Crick, if you've got DNA that replicates, it gets turned into RNA, which is your semi-blueprint, and that again codes for protein. And the proteins are the workhorses of the cells. They go and do stuff. Turns out that there's a lot of RNA pieces as well that also go and do stuff, which are increasingly important. But that's essentially what you've got. If you break anything in the DNA and it codes for something functional, you will change what is happening in the cell. And you will change it permanently, because any other cell that divides from that retains that change, because it replicates from what you've got. So if you break that, everything else after that will look exactly the same. So it's a permanent change. So let's imagine how we could modify this. So if we've got a code of DNA and DNA is read in triplets by amino acids in these sort of codon sort of formats, that would be a code. That's probably it representing a fairly bog standard protein. But if you change the DNA by adding or removing one nucleotide in the DNA, so let's say that little one over there, what you get is, because it's read in triplets, you get this shift. It's now out of sync. And when that sync shifts like that, your actual reading frame changes and will start, instead of a stop codon coming right at the end of the protein, it'll come up really early. And in essence, you've destroyed that gene. So all you need to do is have a single base pair insertion or deletion, if you want to stop a gene from working. So, Genome Engineering 101, now that you know everything about genetics. Genome engineering uses a lot of different tools. We call them molecular scissors. A lot of them have been around for a really long time. Zinc finger nucleases were actually developed in the 1990s. Talons came around in the early noughties, and then CRISPR, the sort of new kid on the block, came around in 2011, 2012. But essentially, they all do the same thing. They create a break at a specific sequence. And we, in the way that we design these scissors, dictate where in the genome we want that break to be. Now, why on earth would you want to break DNA? You want to change it, yeah, but why would you want to break it? Well, it turns out that when you break the DNA of a cell, you create this double-strand break. You actually create, I always think of it as like a tethered rope, and you've got these sort of fluffy ends at the end. The cell freaks out, can't handle a double-strand break. It has two decisions. It's either going to kill itself, apoptosis, or it fixes it very, very, very quickly. And it's got two ways that it can fix that piece of DNA. The one is called non-homologous end joining. Don't worry about that too much. But essentially, imagine it to be like the disorganized first round of cavalry that comes in and just goes and rushes towards that double-strand break, and it just says reseal. If you make mistakes, I don't mind. It doesn't matter what you do, just get those two pieces back together before I kill myself, essentially, in this little cell. So what happens when it does that is that it does make errors, makes mistakes. And sometimes it'll add a few nucleotides, and we call them insertions, or it'll delete. We call them deletions. So we call these indels. So remember when I was talking to you about that frame shift? If you can make that indel not a factor of 3, a multiple of 3 rather, you can stop a gene from working wherever you want. You can do it anywhere in the genome. Okay? But that's kind of the slapdash method, and you're kind of hoping that things are going to work. And they largely do, and it's an incredibly valuable tool. But the other way that you can do it is that you can actually say to the cell, okay, whoa, that's fine, we'll create the break, so your scissors still cut in the same place, but this time you say to the cell, here's a piece of DNA, now I want you to fix that break like this. Add this nucleotide, or add this mutation, or delete these few, or add this whole gene. Whatever you want to do, if you give the cell a piece of donor DNA, a donor template, you can change the genome into anything that you want. There's some limitations. Size is a problem. I think the largest insertion they've done is 7 kilobases, 7,000 nucleotides. The largest deletion is about 100,000 nucleotide. Deletions are oddly enough easier. But to make a specific break, it is much harder, but you can do it. It's much less efficient. But can you imagine, if you know what the mutation is, you can fix it for anything. Now, up until a few years ago, sorry, let me just go back here. I mentioned zinc finger nucleases, talons, and CRISPR. Up until a few years ago, there were probably, I would say, a handful of labs in the world who could do this, because the actual scissors are so hard to make. But now with CRISPR, if you've even got the very basic molecular biology skills, you can do it. So let's just see how this would work with the repair. The way I like to think about genome engineering, and again, this is an important concept and how it's distinct from traditional gene therapy, is that with traditional gene therapy, we'd send in a piece of DNA, but we didn't know where it was going. Genome engineering is a little bit like saying, here are the GPS coordinates, and with editing to make the correct repair, you're saying, here are the GPS coordinates, and here's the cargo that goes with it. Okay. So just to reinforce the way that you would do it, it's literally a pair of scissors, please forgive the get a PowerPoint presentation skills, animation skills. So you cut. Then you provide the cell's repair machinery, then you add your donor template, and it seals it back together. And you've changed one mutation into whatever else you want it to make. Okay, so anything that you want to do. So just very briefly, and this is not too important, but I cannot even tell you how much this has changed the lives of the average molecular biologist. Zinc finger nucleases were made in the 90s, incredibly hard. Talens a little bit later, came from a plant bacteria. And this little guy came out. End of 2011, 2012. Jennifer Doudna and Emmanuel Charpentier, who will win the Nobel Prize soon, we're doing some very basic science research, and this is a reason why people should fund basic science, because they were trying to figure out how bacteria protect themselves against viruses, just how bacterial immunity works. And they came across this little technique. And now all I need to do when I want to use their system, when I want to break a specific part of DNA, because they discovered this little very important protein called Cas9, and they discovered this little structure of RNA, is I email a company down the road from me, and I'll say, can you just make these 20 nucleotides for me, please? And I put them done. So it takes about 3 days to make this. So yeah, that was Doudna and Charpentier. If anyone's interested, you should follow the ongoing battle between Berkeley and the Broad. Feng Zhang, who subsequently, after the ladies discovered the basic method of this at Berkeley, then took what they'd made and very correctly then put it into human cells, despite the fact that Doudna and Charpentier had written at the end of their paper, this could then be put in human cells. So it was exactly there. Everyone knew what was going on. And because of that, and you must make up your own mind about who should have got it. You can maybe be able to tell this slight bias from me. But they filed their patent in 2012, and it was going through the normal processes. But Broad is powerful and very strong. They've got Eric Lander and a couple of other really incredible people, and they got their patents to be rushed through. So they were awarded the patent. And there's been a battle going on between the two. Whoever gets this will control molecular biology for the next, 50, 100 years. So it's interesting. Okay, so now you know how genome engineering works. So there are a couple of things just to reinforce. We can make those indels, those little breaks, and you can directly knock out a gene function anywhere that you want. You can also repair. It's harder, but you can give that donor template and you can say, go and fix the piece of DNA exactly like I want you to here. I've given you the instructions on how to do it. And then there's actually another super cool little method. It's harder, but it's quite sweet. Every person in here, on your chromosome 19, in the middle of a gene that we still don't really know what it does, there's this thing called the safe harbor locus. I love it when scientists get quite cool and groovy about the way they name things. And the safe harbor locus is a site that AEVs, it's a type of virus that we're all infected with, preferentially integrates in. It doesn't do anything. But that's how we know it's a safe harbor locus, because we know that viruses often integrate, and this virus often integrates there, and the DNA is fine with it. It doesn't disrupt any gene or do anything else. So as soon as people in gene therapy and genome engineering realized that this was a safe harbor locus, this could then also be a region where you could insert a whole nother gene if you need to do replacement. And you need to put it somewhere in the genome that's going to be safe, where you're not going to disrupt or activate an oncogene or do anything else. So you can put it in there, the safe harbor locus. Also super cool. Okay, so those are the different things that you can do with genome engineering. There are some issues, though. The main thing is that CRISPR is super easy, TALEN slightly easier, zinc fingers super hard. And as a result of that, CRISPR is a lot more efficient. And you're wondering, why does it have to be so efficient? Just get it done, just screen more, whatever you have to do. But I'm the one who's doing the wet bench work, and I only want to screen 100 clones, not 1,000. And then the last thing that's quite important is that when genome engineering scissors target a specific piece of DNA, what actually happens is that for example, the zinc fingers, I don't know if you remember that like umbrella shape, they actually use a lot of DNA to then bind. The more sequence you have, the more specificity, the more specific it'll be to that part of the genome. CRISPR only means 20 nucleotides. The genome is billions of nucleotides. 20 nucleotide sequence is going to appear a couple of times. And that's the problem with CRISPR. And remember that, because that's going to be the final topic of conversation we have tomorrow afternoon. And that's why there's the big hoo-ha going on, is because we can predict to a degree where else it's going to hit, but we can't, unless we sequence the genome of every single experiment we do, we cannot guarantee what else we've hit. And that's key. That's the biggest problem with genome engineering at the moment, is off-target effects. We can't control it yet. Right, you know everything about genome engineering. That's it. Okay, now it's a very powerful technology, and so that's great, but the problem is that it's not very powerful unless you put it into something that actually has an effect. So the most powerful cell type to use when it comes to this are stem cells. Okay. Now, I know you all know everything there is to know about stem cells, but just bear with me. I'm going to take you back about 70 years to a guy called Warrington, who was a biologist in the UK in the 50s. And he came up with my favorite diagram in biology called Warrington's Landscape. I kind of call it the pluripotency luge. If you are a ball at the top of a series of mountains and valleys, your fate has not yet been determined. Where can you end up? If you're at the top, you might land up here, you could land up here, you could land up here. You have all these options. You don't know where you're going to end. If gravity is the only thing pulling you down, then just depending on how far you go down and which, for whatever, some small microscopic event at this particular place pushes you this way instead of this way, and you land up here, your fate to a degree has been determined. You still have two options. You can still become one of those two, but you can't land up at the other valleys, right? That's how stem cells work. It's exactly the same. A pluripotent stem cell, like embryonic stem cells and the induced pluripotent stem cells I'm going to talk to you about, are at the top, okay? Very powerful. They can become anything. Their cellular fate has not yet been determined. Adult stem cells are different. They are multipotent, not pluripotent. They are where the arrow was. Their fate has been determined to a degree. So when you read about somebody saying they took hematopoietic stem cells and they're going to put it into the brain to create neurons, it's not possible, because hematopoietic stem cells are down this alley and neurons are made from a different lineage. You can't do it. Okay? That's how you'll know when it's dodgy. So this is the power of genome engineering. So I've just said to you, you start at the top, that's the pluripotent stem cells, and you have your adult stem cells, and the cells at the bottom are your differentiated cells. So a skin cell, or a terminally differentiated blood cell, or a heart cell, or anything you can think of. They're at the bottom, and they're done. Mostly they don't divide, but a few of them do. So I've just said to you, that is the dogma. That's how it works. It turns out that that's not true. So again, in the 60s, John Gurdon he looked at a frog, and he was like, so it's a bit confusing, but if I take the DNA from a skin cell of a frog, I take that DNA out, and I put it into the egg cell of a frog, I can make a whole frog. So the DNA contains all the information. So what is it that's in the egg cell that makes this different from the skin cell? We learned then that the DNA contains all the information for every single cell in the body. Then, in 2006 and 2007, Shinya Yamanaka and his postdoc Takahashi were working on a way of trying to take Gurdon's information and try and find a way of working out which genes were responsible for this shift from a terminally differentiated cell to a stem cell. And they got it like on almost their first try. And essentially, what they did, which later gave them the Nobel Prize in 2014, is they worked out that if you added just 4 genes to a skin cell, so if you've got some freckles, I've got here, you just need a biopsy, skin biopsy about that big, the size of a freckle. Those skin cells will grow out in a dish. And if you add 4 genes to those skin cells, you can turn them into a pluripotent stem cell that acts like an embryonic stem cell. Now what that means is that I could make pluripotent stem cells from every single person in this room if I wanted to. And now that is a pretty amazing tool. So let's go back to the different cell types. Embryonic stem cells, they're at the top of Warringtons Landscape. So we can make, with those embryonic stem cells, when Jamie Thompson first did this in 1998, basically since then, for the last 20 years, scientists have been using these embryonic stem cells to figure out how you can turn them into brain cells, or turn them into blood cells, or turn them into whatever else, all in a dish. So that's the amazing thing about embryonic stem cells. They obviously come with a problem. Well, they come with a number of problems. The main one being ethics. Okay, depending on which side of the fence you are, it creates an issue. But they're very, very powerful. And in fact, so much so that people like Lawrence Studer, who thinks at the Broad as well, actually, has been optimizing how to make Parkinsonian neurons from them for the last 10, 15 years. And he can make in a dish like 95% dopaminergic neurons from those embryonic stem cells, which is pretty phenomenal, actually. So let's go back to induced pluripotent stem cells. Embryonic stem cells, one option, potentially there, but a couple of issues. The reason why it's such a big deal is because, like I said, essentially you could take from any individual in this room, you add your cocktail of genes, we now call them the Yamanaka factors, and you can turn them into patient cells. Now that's super cool, because the one thing you could do with this is that if these cells have a recognizable genetic defect, I can turn them, I can repair them, correct the difference, turn them into the cell type that I need to replace them with, and stick them back into the person. And all I needed to do was take a skin biopsy. So this could potentially start working in the favor of neurodegenerative disorders, heart disorders, liver issues. We could make piles and piles and piles of the right tissue, and they are autologous. They come from you. There will be no immune rejection. You could have your own bank set up, just waiting for you. And you don't even have to turn them into all those different types of stem cells at once. You just have to salvage the skin biopsy at some point. The other option, again, this is where we work on, is that you can make these cells, patient-specific cells, turn them into the cell type that is affected by the disease, but you keep them in the dish. So like Studer's Parkinsonian neurons, he wants to figure out which drugs make them revert back to a normal neuron. So he has normal neurons, and he has these neurons that are sick, and now he can use them as a drug screening tool on a person-to-person basis. And that's largely where our area of work is. Okay, so that's why induced pluripotent stem cells is so cool. They're also pretty cool is because they're so easy to work with. So this is where I get to show off just a little bit. So we've made a lot of neurons in the lab. You can actually see the cell nuclei sit here, and they sit in a clump, and then you get this beautiful sort of dendritic arborization that comes out. The neurons grow like wildfire in the dish. We also make these. I don't know if you're going to be able to see them. If you focus your attention here, can you see those two little spikes? This is the type. Give it a second. Can you see them to that? Anyone want to guess which cells these are? The macrophages, the white blood cells. So the macrophage to eat. So they're terribly sweet actually, because we leave them out for too long. They get a bit cold and then they slow down. So they stop moving after a while. So we can make those. And just to give you an idea of the extent of how much they move, see how they talk to each other. The eye in blue, and then you can see the pseudopods walking around trying to pick up other little things, and then they talk to each other. So they're really, really sweet. And then we also play around with these as well quite a bit. Anyone want to hazard a guess? We do this. Should be going about the same size. So we can grow this for up to a year in a dish if I remember to feed them. Which our students remind me to. But they're super sweet. They also slow down if they're kept out of the incubator for too long. So the point being is that this is the kind of technology that we've established here, and they're due to GCT as well. This is not hard. Making stem cells from any person in this room, and those are just 3 of the cell types that I've shown you we could make in a dish. Okay. So let's to what in for a second. So I've shown you what pluripotent stem cells can do, embryonic and now induced pluripotent stem cells. And the reason iPSCs are so important is that it's an alternative from embryonic stem cells. So it allows us to do the same work without creating too much of a problem. But I do also need to mention adult stem cells because they are an incredible, incredible option. So it turns out that you actually have stem cells in every single, I think almost every single tissue in your body. You have stem cells in your brain, in your muscle, in your heart, in your liver, in your blood, skin, digestive tracts. You have stem cells everywhere. So the beauty about using these things is that you can actually now take from each person in here, you can extract those adult stem cells, and you can try and grow them up in a dish, and then put them back in if need be. The only problem is that when you do that, you actually modify them a little bit. They don't really like growing on plastic. And I'll show you why. Please forgive me. I'm going to go forward 2, and then I'm going to 3 or 4. Let me just see if it's here, and then I'm going to come back. Okay. So this is the reason why it's a problem. This is a karyotype of a cell. So when you're growing cells in a dish, karyotype is the number of chromosomes that you have. So you should have 23 pairs, okay? And almost every single cell, every single cell in your body, you should have 23 pairs. And that's what they look like if you stain them with a special marker, and they look really pretty. Has anyone read the book Henrietta, The Immortal Life of Henrietta Lacks? Yeah, that's what hers look like. So these are cells that have been growing in culture for 60 years. You don't have to be a cytogeneticist to notice that you shouldn't have 5 copies of chromosome 1. And I'm not entirely sure what's going on with chromosome 10, but they refer to it as chromosomal shattering. This is what happens if you grow cells in a dish for too long. So again, that's why you have to be really, really careful about growing adult stem cells in a dish and trying to bulk them up and make as much as you can. They don't like the plastic, and they start picking up mutations that make them grow faster. And mutations that make cells grow faster are cancer mutations. Okay. So this is the balance that scientists have to work with in trying to figure out which cell types they want to do. So let's just go back here. So I've spoken about the different cell types that you can use, but there's also this incredible new aspect of technology, which is using scaffolds. Most of this work I've spoken about will either be about cells that float in the dish, like the blood cells, or a lot of the rest of them are actually stick to the bottom of a plastic dish. And it turns out that that's actually not really good for them. What you've got to do is give them a 3-dimensional shape. Because as soon as you give them a 3-dimensional niche, they start acting more and more like the actual cells that you have in your body. And remarkably, if you actually let them do it themselves, they self-assemble. It's like they know exactly what shape they're supposed to be making in the dish. So this is probably a combination of self-assembly and manipulating a 3-dimensional system, but specifically talking about how engineered stem cells can make such a big difference as we start launching towards structures and organs. A little girl had a severely damaged iliac vein. So for those of you with the medical mind, it's this one over here. And the iliac vein is pretty simple. If you actually sort of slice through it, it's made of some very simple layers. It's got its outer layer of collagen fibers, okay, so that's not cellular, it's fibrous. And then there's a smooth muscle and an endothelial layer. So only 2 cell types required to make this. So what the doctors did, which was super cool, is that and an individual who donated some of his organs, they took his iliac vein and they decellularized it. You wash it through a number of different cycles of different pHs and salts and whatnot, and it removes all the cells. And what are you left with? A scaffold, a structure, okay? Now you put that structure into the same kind of thing that you find at a rotisserie chicken kind of vibe. You see it here, and they actually sort of rolled it, kept it rolling for a period of a couple of weeks. In the first few weeks, they added the smooth muscle cells, which attached and formed that layer. And once they'd done that, they then added the endothelial cells and added the subsequent layer. And that's a fully functional iliac vein. She's now bouncing around doing gymnastics. So while I've gone on and on about induced pluripotent stem cells, adult stem cells where you can get, and even more terminally differentiated cells where you can get this, you can actually do quite a lot with them. if you do it relatively quickly in terms of structures that you can create. So there's also a massive area into 3D printing. I'm actually not going to go into it because it's just such a huge aspect. But I do just want to bring it up. There was I think they've now done 10 cases. If anyone's really interested, Antonia Tyler does the most beautiful TED Talk on 3D printing of, I think it's a kidney and a bladder. They've done 3 bladders now, transplanted those, made them, and added and surgically added those to individuals who've now subsequently done incredibly well from this. So that's a huge area of research. One of the problems with organs, though, and we still haven't quite figured this out, is that a bladder is one thing. But remember, every single cell in the body must be lying next to a capillary, a blood vessel of some kind. So it's complex. It's not just a single line of cells or then another layer. There's this intricate network that must grow with it and be folded through it, so that all of those cells have access to oxygen and food, really. So one of the really amazing areas of research that comes into engineering stem cells are now hydrogels. And what they're doing is spending a lot of time, and this sounds weird, figuring out the consistency of the jelly that you would need to allow for natural capillaries to create this perfect vascular network that could then be used in conjunction with the 3D printing work. So that's where we are with that. So we've been through why you shouldn't stay in culture for too long. And I think you're with most of the Warringtons Landscape. So the thing is with the stem cells is that if you're at the top, you get a lot of numbers. You can make any cell type in the body. You can't add the stem cells as they are. This is really important. They will cause cancer. So that is a problem. Whereas at the bottom, you don't have so much of a tumorigenicity point of view, but the cells don't last forever because they don't divide. So you've actually got this problem because you want lots of them, but you don't want them to give you cancer. So this is the balance. So remember that thing I said to you at the beginning. Why is genome engineering with cellular treatment so complicated? These are the considerations that people have to make for every single aspect. So the other thing that's really important, quite an interesting moral discussion, is whether or not we should be creating private banks of cells. So remember I said every single person in this room could have their own autologous cell stored. But why do that? Why not have an allogenic bank? So there are groups of individuals in this room who allogeneically match. You're immune matched, the same way that you would match for bone marrow transplant or whatnot. So you could actually bulk up enormous numbers from a few individuals, which would then apply to everybody. And then each one of those source of cells could be carefully checked to make sure that they don't have the wrong mutations in them. So that's an option. I've spoken also about this balance between you want the cells to take in the body, but you don't want them to overtake. So you've got to be able to add little control mechanisms. And perhaps one of the cleverest systems that they've done is they've actually added a suicide gene. So with Leila's treatment, remember, first they had to add the CART to make sure it recognizes cancer. Then they had to break the gene that would be recognized by her body. And then they added a third one in the most updated version. It's a suicide gene. So if they think the cells are taking over her body and there's some kind of immune problem, you add a bog standard mammalian antibiotic, boom, and they all die, without any impact on the individual concerned. So those suicide genes are actually quite clever. So what I want to, as we're heading towards the end, I want to just talk about delivery. I've spoken about how you can take adult stem cells, you can repair them, and you can put them back in the body, okay? I've told you about this amazing technology of making induced pluripotent stem cells, correcting them, and you can put those back in the body. Then there's also the aspect of direct in vivo editing, and this is actually cell-free. This is just using the genome engineering tools on their own. And when it comes to this, the issues of how to do this are effectively the same as they are for traditional gene therapy. Because here's the thing. It's effectively a piece of DNA, right? How do you get it into the body? That's what you're chopping it up. So it's all about delivery. And I think the best way to explain this is an analogy with what I like to call a de-weaponized tank. So a virus is like a tank, okay? It can go anywhere, It's incredibly dangerous because it's got these things that will blow you to bits or whatnot. So what you can do, though, is you can say, okay, that's fine. What we're going to do is we still want your ability to go anywhere. We just don't want your weaponized action that could be dangerous. So you do de-weaponize the virus itself. And one of our favorite ones that people have been using is HIV. So HIV is a great example. It can infect a lot of different cells. It can get anywhere, and it actually integrates into the genome. So what you do with that is you take out all the stuff that causes disease, but you leave all the components in there that just allow it to move around and go anywhere. And then instead, what you then do is you take your CRISPR scissors, for example, the DNA that codes for that, you put that into the virus, and now that's your de-weaponized tank. It can go anywhere, but now it's delivering not cells or whatnot, it's delivering something that will rescue you. So that's essentially how delivery works for most of these. The difficulty is that depending on where in the body you need to take up these genome engineering aspects, makes it difficult to know how much of an effect you're going to have. So largely, and most of the advanced trials are blood diseases, so systemic. You can actually just do a systemic injection, and that's already fairly well established. Neurological disease is complicated. You can do systemic, but the problem is the blood-brain barrier, so the viruses really battle to get across. There are some that you can make that slip through. And then there are obviously sort of direct injections that are options. The other thing is that you have lots of different tissues in the body, and it turns out different viruses have different favorite tissues. And we now know which viruses these are. So AV type 2 really likes liver. So if you need to fix a liver gene, then you use the de-weaponized tank that likes liver. And you can make that specific for every single tissue that you work on. The ex vivo aspects of delivery are a little bit more complex in terms of the fact that if you're going to use adult stem cells, remember Warrington's Landscape, if I want to have an impact on neurological disease, I can't use hematopoietic stem cells from bone marrow. There's no point. So you have to choose the lineage that's going to make the cell types that you want. Induced pluripotent corrected clones are a relatively easy way to go, but there are, at the moment, still some issues with how we're going to actually get those cells into the right place. So there's a trial going on in Japan now to work with retinal pigment epithelium, and they're actually almost creating it's like a glue-like paste, and that's where the cells are going. So delivery is still at the forefront as an issue. So let's just talk about very briefly some examples. So Leilas, just to reinforce how they did the genome engineering for her, and this would work for a number of different leukemias. So here's the original T cell that she had, unmodified, and this can come from any healthy donor. So anyone can donate these, right? Then what they did, they inserted that chimeric antigen receptor that specifically goes for exactly the cancer that she has. And it doesn't just have to be this cancer for CD19, it's a PD1, it's a number of different other things. As soon as you can recognize what that cancer has, you can use this method to target it specifically. And then they added their suicide gene, and then they disabled their recognition gene. And that is how Selectus has developed their FDA-approved UCART19, a fully modified cell that would directly target her cells. And there are a number of other different examples. So let's talk about other things that have been going on. So HIV, oddly enough, is also at the forefront of genome engineering. So a few hundred years ago, when the plague hit Europe, not Africa, it selected for certain individuals that would survive, who would be better off, okay, without the complications of the disease. Fast forward a few hundred years, and when the French groups were finally making the connection between HIV and AIDS, they had obviously massive numbers of patients and controls, right? And that's how they were able to pick up the connection. But they also had a very small group of individuals who, if they looked hard enough, were positive for HIV, but 10, 15 years later, no clinical symptoms of AIDS. We now know them to be elite controllers. And there are probably about 10% of you in this room who are elite controllers, who are certainly carriers of that gene. And what turned out to happen is that during the plague, there's a mutation in this gene called CCR5. So if you're a white blood cell, for example, and HIV needs to infect you, it needs two things. Think of it like a Sputnik. So it has to hang on to this one thing called CD4, and then it has to hang on to CCR5. Once it attaches to both, it's stable and it can inject into the cell. Elite controllers only have CD4. Their CCR5 is broken. So HIV kind of, you can imagine it just like, it can't infect. So if there are people walking around who are naturally refractive to HIV, and they're not completely fine, and I want to bring this up because we'll talk about it again tomorrow, but this is a potential target for a permanent functional cure for HIV. And the only reason it's in 10% of Caucasians and not in people of African descent is that the plague didn't hit sub-Saharan Africa. So it was selected for. As it turns out, African individuals are actually elite controllers. We just don't know what they have. They don't have CCR5, so there's something else. So in 2008, when Timothy Brown had developed a type of leukemia, he was HIV positive and had been on antiretrovirals for some time. His doctor said, okay, well, if we're going to have to do a bone marrow transfusion, let's not just do it from anyone who matches. Let's pick someone who's CCR5 negative, who's got this mutation. And they did that. And 10 years later, he is the only person who's been functionally cured of HIV using the natural mutation. So I'm aware of the fact that people are going to sit in this room going, what, we're going to do genome engineering and bone marrow transplants for every single person in sub-Saharan Africa with HIV. I'm aware of that. But consider what we're doing in the background. Consider lifelong antiretrovirals. Consider adverse drug reactions, which we're going to talk about tomorrow as well. It's just an idea. So that's where most of the advanced trials are ongoing, are CCR5 in the States. CART for cancer, and then also, and this is really wonderful because these are the diseases that people have been working on for a really long time. We've known what the gene is for a long time. We've known what happens. These are terrible disorders for someone to experience, for families to go through as they track through their families. And now finally there is actually an option. So one of the really cool papers that came out earlier this year by Frederick Olson's group was that they actually were able to do for muscular dystrophy, they used genome engineering CRISPR, the in vivo option, so directly systemically injecting it into canine models, and restored the dysfunctional dystrophin to nearly 90%. That's a cure for dystrophin, okay? Particularly close up to 50% in heart muscles, and that's the reason why these boys die and with Duchenne muscular dystrophy. I'm going to talk about organoids tomorrow. They're really sweet. But for cystic fibrosis as well, this is an option. So if you take stem cells from the intestine of a cystic fibrosis patient and you grow them up, they kind of look like this, make these little spherical organoids. And if you add a certain calcium stimulus, they just kind of sit there. But if you genetically correct them, they do exactly what they're supposed to do and react to the calcium in a dish. Again, this is a disease in a dish model. Imagine you could test out millions of drugs to figure out which drug can restore the function for these patients. So, with that, going back to that original figure, okay? Now you're starting to get an idea of the kinds of things that you can do with in vivo therapeutic approaches, directly adding the actual tools, the scissors, okay? You can also start getting an idea of what you can do with adult cell editing and induced pluripotent cell technology. And I've started just mentioning a few of the things that we can do by modeling disease in a dish using these tools. So with that, I'm going to end for today, hoping there are some questions. Tomorrow is actually with all the juicy stuff. We can get onto ethics and things like that. But if there are any questions, now would be a good time to ask. Oh, you mean use it as a weapon? I think smallpox has got its own way of doing that, but It's hard. So you don't need genome engineering to modify a virus. We've been able to modify viruses since 1963, since Paul Berg. So it's probably not the right thing to tell you because it's even worse than what you were expecting. Recombinant DNA technology has been around for long enough for people to have been able to, if they wanted to, do that. Yeah, you don't need genome engineering to do that. Yeah. Put a bit of a dampener on things. Anything else? Yeah.
When couples go for genetic counseling and something problematic throws up, can you then say to them, but it would be all right if we?
Yes. So I was actually going to bring this up tomorrow during the ethical discussion. With genetic counseling, if you know what the mutation is, let's say if that's the example, so you've now tracked something and they want a baba, but they know that they've got 50% chance of carrying. Let's say it's a dominant, autosomal dominant, and they don't want one of those hits. They can do IVF, and then they can do PGD. So PGD is called pre-implantation genetic diagnosis. And so my personal, I'll discuss this in more detail tomorrow, particularly given the latest hoo-ha with China that's just happened in the last month with the little gene-edited baby girls. But the difficulty is, if you know what the mutation is, PGD is a way of choosing the healthy embryo without having to do any genome engineering. To date, I've yet to come across a single person who can give me an example of why you would need to do genome engineering in an embryo. There isn't one. I'd be delighted if someone could come up with it, but we do this with the students all the time. Mitochondrial disorders, there's 3-parent babies now, so you don't need to do that. I can't think of one because you can always choose a healthy embryo. So that's the problem I have with it in embryos. Having said that, they're doing fertility research in the UK. They've got permission to do it, but they stop at a week. You're not allowed to go further than a week. I don't see any point. It's amazing technology for what else we can do, but yeah.
What's the potential of cryogenic freezing your vegetable as a source of stem cell
So there are at least 2 companies in the country that do that already. Cryosave and was NetCells and now NextBio. And there are numerous companies elsewhere in the world. It is feasible. It is scientifically plausible. I have two issues with it. The one is that, and the main one is that the amount that you get, so you get mesenchymal stem cells from the cord, and then you get hematopoietic stem cells from the cord blood. Okay, I think the cord blood is about 5 mils. I think maybe 15. It's not a lot. Now, you're not getting a lot of cells in there, and they are frozen, and to the best of my knowledge, if you freeze them correctly cryogenically, they can be brought back up, but you don't have enough. So the last I looked into it, and my last understanding of it was that there's enough to do a single infusion for a child under the age of 12 with a rare type of acute lymphoid leukemia, and that's been done once in the world. That's not to say that one day the technology will then figure out a way to grow them up. The other thing is that I believe it shouldn't be a private bank. It's my own personal opinion. I think it should be a public bank. Because if it's a public bank, you're now combining all the allogenic samples together, and now you really have bulked up amounts to which people could actually genuinely get a great deal of benefit, in my own opinion. But it's not scientifically, it is feasible, and certainly theoretically could be of great benefit in the future. Yeah.
treating somebody. If you're going to introduce these big
Yeah, I mean, that's the problem, right?